Alright, yes, assalamu alaikum everybody. How are you all? I hope that you all are doing well. Welcome back to my new lecture of cell structure topical MCQs. Today we are going to be discussing about the cell organelles and their description. Like in the previous two videos, we discussed about cell microscopy. We discussed the light microscope, the electron microscope, right? In today's video, we will be discussing about the cell organelles in their description, the MCQs related to that basically, right? So let's move on to question number 1. It is a very lengthy question, but it is easy if you know about the organelles and if you know about their description. Question number 1, it is from October November 2020, paper 1 to question 4. It over here says that 4 students were asked to match the function with the appearance of some cell structures in the animal cell. Alright, so you have to note over here that they are talking about the animal cell. The functions were listed by number. mRNA passes through to the ribosome, synthesis of lipids, packaging of the hydrolytic enzymes that will remain in the cells. We know that the mRNA passes through to the ribosomes. Alright, so if I talk about over here, so we know that the mRNA is basically synthesized in the nucleolus, right? No, sorry. Alright, now they are saying that the appearance were listed by the letter V, W, X, Y, Z. The appearance, basically they are saying that these are the appearance, from it are the appearance of these structures which do these functions, right? Then they are saying which student correctly matches the numbered function with the appearance of the cell structure. So they are basically saying number 1, which is the mRNA passes through to the ribosomes. So the mRNA passes through to the ribosomes via the nuclear envelope, right? You can see over here, this is a nuclear envelope which is continuous with the which is basically enclosing the nucleus, right? Nuclear envelope which is double membrane bound. Over here you guys can see the pores, right? So basically the mRNA, when the DNA is transcribed and mRNA is generated, so mRNA passes through these nuclear pores to the ribosomes. These dotted structures over here are the ribosomes, right? So they pass from these pores to the ribosomes, right? So we have to see that where the nuclear envelope is there. Nuclear envelope, we know that it is a double membrane bound, right? It is double membrane bound and it is interspersed with the pores, right? I also showed you the pores over there, that nuclear envelope has pores inside it. So 1 is basically matching with the description of X, right? So we have eliminated option A and option B over here. Now 2 is the synthesis of the lipids. We know that the synthesis of the lipids takes place in the smooth endoplasmic reticulum, and the smooth endoplasmic reticulum is basically the membranes which surround an enclosed inner cavity, right? So this is the smooth endoplasmic reticulum. This is the description of the smooth endoplasmic reticulum. If I discuss about W, so non-membrane bound spherical structure, these are the ribosomes, right? These are the ribosomes. And if I talk about Y, non-membrane bound cylindrical structure, so these are the centrioles, right? We have two non-membrane bound structures in the animal cell. They are the ribosomes and the centrioles. If I talk about Z over here, so Z is the membrane bound sac arranged as a flattened stack. Acha, if they are membrane bound sacs which are arranged at as a flattened sac, so these are the Golgi body. Yeah, you can say that these are the Golgi apparatus, right? These are the Golgi apparatus. And we know the function of the Golgi apparatus is the collection, packaging of the protein, right? Modification, right? These are the functions of the Golgi body, and they are the one because they pack the hydrolytic enzymes as well, they pack the protein as well, and proteins are the enzymes. So the packaging of the hydrolytic enzymes is done by the Golgi apparatus. So 3 is basically Z over here, right? And 2 is, I told you guys, V over here. So 2 is V and 3 is Z. So hence the correct answer over here is option D, right? I also put an image over here to show you guys the nuclear envelope, that this is a nuclear envelope which is double membrane bound. This is the nuclear envelope which is double membrane bound, and it is often continuous with the rough endoplasmic reticulum. This MCQ will come further as well in the upcoming MCQs, that what is basically continuous with the nuclear envelope. It is the rough endoplasmic reticulum. How do you identify the rough endoplasmic reticulum? By the ribosomes attached to it, right? So by this you can identify the rough endoplasmic reticulum, and these are the nuclear pores. You can see over here, from it the transcribed mRNA leaves the nucleus and goes to the ribosomes, right? I hope that you guys are getting the concept. I hope question 1 is clear to you. Now let's move on to question number 2. Question number 2 is basically saying over here that it is from October-November 2020, paper 1 to question 5. It says that which are found in the typical prokaryotic cells and in the typical plant cells. Alright, if I talk about a typical prokaryotic cell, so you can see over here it is a typical prokaryotic cell and it is a typical plant cell over here. In the typical prokaryotic cell and the plant cell, they are saying that what is found in both, right? They are talking about the common structures in both. So we know that prokaryotic cell has a peptidoglycan cell wall, and typical plant cell has a cellulose cell wall, right? So cellulose cell wall is found in both. Cytoplasm obviously is found in both. Now if I talk about the circular DNA, so we know that in the prokaryotic cells the plasmids are found, right? Plasmids are the circular DNA. So plasmids are found in the prokaryotic cells, right? And in the typical plant cells, we know that there are chloroplasts in the typical plant cell, and chloroplast has the circular DNA, right? And chloroplast has the circular DNA over here. So where is the chloroplast over here? Yes, this is the chloroplast in a typical plant cell. This is the chloroplast and it contains the circular DNA, right? So hence the correct answer to this MCQ is option A. 1, 2 and 3. All the three structures are basically found in both the prokaryotic cells and the plant cells. Over here you can see the prokaryotic cell. This is the flagella, right? The flagella of the bacteria, which helps bacteria to do the locomotion, to move. This is the plasma membrane. Then this is the peptidoglycan. Okay, this is the capsule, the slimy capsule basically, right? And then alright, so in this structure, if you guys can see, so there are two lines. One is of the cell membrane and one is of the cell wall, right? And these are the mesosomes, which contains all the respiratory enzymes, photosynthetic enzymes. These are the ribosomes, cytoplasm. Now plasmids are not shown over here, otherwise they have a circular structure of DNA which are known as the plasmids, right? So I hope that the prokaryotic cell structure is also clear to all of you. Now let's move on to question number 3 over here. Now question number 3 is from October-November 2020, paper 1-1, question 1 over here. It says that which row shows the correct order of size of these cell structures, right? So this is something you have to remember. We know that cell surface membrane, the width of the cell surface membrane is very tiny, right? It's very much tiny. So obviously it would be the smallest of all. So 3 must be the smallest of all. And then chloroplast is larger in size than the mitochondria. You must know this fact. So 4 is larger in size than 1, right? And then the width of the ribosome. Alright, so ribosome is smaller than the mitochondria, right? It is smaller than the mitochondria but larger than the cell surface membrane. So it comes over here. So hence the correct answer for this MCQ is option C. The largest width is of chloroplast, then comes the mitochondria, then the ribosome, and then finally the smallest width is of cell surface membrane, right? So I hope that this MCQ is also clear to all of you. Alright, so let's now move on to question 4, which is from October-November 2020, paper 1-1, question 5. It is similar question to question number 1. It says, but it is a little different question, right? So let's begin with this question. It says that 4 students were asked to match the function with the appearance of some cell structures in the animal cell. Same, these functions are given and then the appearance are listed. we have to correctly match the numbered function with the appearance of the cell structure, right? Synthesis of the polypeptides, we know that synthesis of the polypeptides happens in the ribosomes. Now we have to look at the appearances and we have to find that which appearances matches the ribosome, right? Synthesis of the lipids happen in the smooth endoplasmic reticulum. Packaging of hydrolytic enzymes, I already told you guys that it happens in the Golgi apparatus, right? So synthesis of the polypeptides, synthesis of the polypeptides is happening in the non-membrane bound spherical structure, which is the ribosome. Double membrane bound interspersed with pores, it is the nuclear envelope. But we don't have it in the functions, right? Nuclear and the functions are not related to the nuclear envelope, so obviously we are not going to count it, but we are going to count for the synthesis of polypeptide, which is W, right? So we will eliminate C and D over here. 2 is the synthesis of lipids. So I already told you the smooth endoplasmic reticulum description is the membrane which encloses the inner fluid, inner cavity, right? So smooth endoplasmic reticulum is V. 3 is the packaging of the hydrolytic enzymes, which is the membrane bound sacs arranged in the flattened sacs. These are the Golgi bodies, right? So Z. So hence the correct answer over here for this MCQ is option A. If I talk about structure Y over here, so we know that structure Y is the centriole. Centrioles are also non-membrane bound and cylindrical structures. So I hope that you all got question 4. Now let's move on to question 5 over here. Alright, now so question 5 over here is basically from October-November 2020, paper 1 question 6. It says that the DNA of a typical prokaryotic cell is naked and circular, right? It is circular and naked. Naked means that there is no nuclear envelope, right? There is no nuclear envelope. And then it says that which statement describes how the DNA of the eukaryotes differ from the DNA of a typical prokaryotic cell, which is the bacterial cell, right? Only the DNA of the eukaryotes has a nuclear envelope around it and it is double helix. Okay, only the DNA of a eukaryote has a nuclear envelope around it. Till here the statement is correct, but and it is double helix, this part of the statement is incorrect. Why? Because we know that eukaryotes, whether they are eukaryotes or whether they are prokaryotes, the DNA remains double helix. DNA remains double helix. So it is not particularly for the eukaryotes, right? If you read the question carefully, they are saying which statement describes how the DNA of the eukaryote differ from the DNA of a typical prokaryote. So this is not a differing point, the double helix part. Only the DNA of a eukaryote has the nuclear envelope around it and is circular. No, the DNA in the eukaryotic cell is linear, right? Only DNA of the eukaryotic cell has protein attached to it. Yes, this is correct. If you know that if you know about the proteins, so they are basically talking about the histone proteins in the chromatin threads, right? And is a double helix. Again, the double helix thing is incorrect, right? Only the DNA of the eukaryotes has a protein attached to it. Yes, it is correct. We know that we have like this is a chromatin thread, for example. For example, if this is a chromatin thread, so we have some histone proteins attached to it, right? Which makes up the nucleosome. I already discussed it in one of my video, nucleosomes, right? So only DNA of the eukaryotes has protein attached to it and is linear. So hence option D is our correct answer over here. Only the DNA of the eukaryote has proteins attached to it and is linear. I hope that this is clear. Alright, so now coming to question number 6, which is from October-November 2020, paper 1-3, question 4. It says that which part of the cell is often continuous with the rough endoplasmic reticulum. So I already told you above that this MCQ will come later, that which part is basically continuous with the rough endoplasmic reticulum. So you can see over here that this is the rough endoplasmic reticulum and you can identify it by the dotted structure of the ribosomes which are attached to its surface, right? These are the dotted ribosomes attached to it. So it is the rough endoplasmic reticulum, which is basically continuous with the nuclear envelope, right? This is the double membrane bound nuclear envelope, and the rough endoplasmic reticulum is basically continuous with the nuclear envelope. So hence the correct answer for this MCQ must be option D over here, nuclear envelope, right? The rough endoplasmic reticulum is continuous with the nuclear envelope. I hope that this was easy for you guys. Now let's move on to question number 7. Question number 7 is from October-November 2020, paper 1-3, question 5. It says that which statement about ATP are correct? We know that ATP is the energy currency of the cell, right? Energy currency of the cell. It is the primary source of the energy in our body. Then statement 1 over here says that it is produced in the chloroplast. No, we know that ATPs are produced in the mitochondria only, right? ATP, the production of the ATP is the function of the mitochondria because it is the powerhouse of the cell. It is the powerhouse of the cell, right? It is used during the protein synthesis. Yes, obviously. Why? Because protein synthesis is an energy consuming process. Energy consuming process, and ATPs are used during the energy consuming process. It contains deoxyribose. Absolutely incorrect. Why? Because we know adenosine triphosphate. It contains 3 phosphate group. It contains an adenine nitrogenous base, and it contains a ribose sugar, which is a pentose sugar, right? So hence statement 3 is incorrect. Then it says that it is used in the facilitated diffusion. Incorrect. Why? Because facilitated diffusion is a passive energy, it is a passive process, right? It is a passive process. If it would be written over here that active transport, so it would be correct, but it is written that facilitated diffusion, so it is a passive process. It doesn't use any energy. It is used to load the sucrose into the companion cell. Yes, absolutely correct. Why? Because we know that the loading and unloading of the sucrose into the companion cell is a active process. Alright, so there is a little mistake in the question. I'm so sorry for the misunderstanding, everyone. Basically, the ATP is produced in the chloroplast. Now, how the ATP is basically produced in the chloroplast? ATP is produced in the chloroplast during the photosynthesis reaction, right? During the reaction of the photosynthesis, the ATP is produced. Now, if in your A2s, you are going to study that photosynthesis is divided into two parts, the light dependent reaction and the light independent reaction, and in the light dependent reaction the ATPs are produced. For now, you just have to remember that photosynthesis is involved in the formation of the ATP as well, right? Photosynthesis gives you ATP as well. So you can say that the correct answer over here would be option A, 1, 2 and 5. All these three statements of 1, 2 and 5 are correct over here. Alright, so now let's move on to question number 8 over here. This question number 8 is from May-June 2020, paper 1 to question 3. It is saying that which row correctly shows a feature of a cell structure. Site of the protein synthesis. We know that the site of the protein synthesis is ribosomes, right? Ribosomes. Rough endoplasmic reticulum is also a site of the protein synthesis. Option D and option A are incorrect. Let's eliminate option A and option D. Makes the lysosome. We know that the packaging lysosomes basically are the vesicle containing the hydrolytic enzymes, right? And these hydrolytic enzymes are packed into the vesicle in the Golgi body, not in the ribosome. So hence option C is incorrect. Our correct answer over here would be option B, ribosomes and the Golgi body. Now let's move on to question number 9 over here. Question number 9 over here is from May-June 2020, paper 1 to question 5. It over here says that the features of some cells and the cell structures that make the ATP are listed. Has an outer boundary membrane and folded inner membrane. We know that the outer boundary membrane and the folded inner membrane is basically the mitochondria, which is somewhat like this, which has a folded inner membrane, right? Folded inner membrane of the mitochondria, right? And there is a outer membrane. So option statement 1 is basically describing the mitochondrion. They are asking that which row identifies these component, right? So the identification of the one is basically the mitochondrion. Then the statement 2 says that has peptidoglycan cell wall outside and outer boundary membrane. So we know that this is, if this is a bacterial cell for example, so it has a peptidoglycan cell wall as well, right? So it is a prokaryotic cell, or you can say a bacterium, right? And then in statement 3 they are saying that has a double boundary membrane and stacks of inner membrane. I think over here I believe that they are talking about these stacks, right? These are the stacks, basically the stacks of the thylakoid, right? Thylakoid are the flattened sacs, and they are basically arranged in stacks which makes up the granum, right? This hole is known as the granum. This hole is known as the granum. If I explain you over here, so basically we can explain it in a way that thylakoid are the flattened sacs, right? And they are joined together to form the sacs of the thylakoid, which are known as the grana, right? And then they are interconnected to each other by the lamella. These are the lamella, which are the membrane, basically they are interconnected by the membrane called the lamella. So they are talking about the chloroplast over here. So hence the correct answer for question number 9 over here would be option D. Now in question 10, which is from May-June 2020, paper 1 to question 6, it says that mitochondria are thought to have evolved from the prokaryotic cells that were ingested by an ancestor cells. Which features have these prokaryotes lost during their evolution into the mitochondrion? Alright, so basically they are over here saying that which features have these prokaryotes lost during their evolution into the mitochondrion. So we know that they have lost the cell wall. How? Because we know that mitochondria is a double membrane bound organelle, right? And it does not have any organelle, and it does not have any cell wall. It do has the ribosome, it does has the internal membranes, it has the circular chromosomes, but however it does not have the cell wall. So these prokaryotes basically have lost their cell wall during the process of evolution into the mitochondria, right? I hope that this is clear. Let's now move on to question number 11 over here, which is from May-June 2020, paper 1-1, question 2. It says that which structures are found in the typical eukaryotic cell. In the typical eukaryotic cell, basically ATS ribosomes, obviously ATS ribosomes are present, linear DNA is present, which is enclosed in a nuclear envelope. We know that. If I talk about the 70S ribosomes, so yes, 70S ribosomes are also present, because we know that in a typical eukaryotic cell, we can find the mitochondria as well, right? And mitochondria has the 70S ribosomes as well as the circular DNA. So hence the correct answer of question number 11 is option A, 1, 2, 3 and 4. All these structures are found in the typical eukaryotic cells. Now let's move on to question number 12, which is from May-June 2020, paper 1-1, question 11. It says that the photomicrograph shows the ultrastructure of a part of a cell. Which statement about these type of cells shown in the photomicrograph is correct? If you identify the structure shown over here, so it is the ultrastructure of the chloroplast, which is clearly indicating the plant cell, right? It means that you have identified it by the help of the chloroplast, that the cell they are talking about over here is basically the plant cell, right? And how do you recognize the chloroplast over here? Basically you can see the stacks of the thylakoid, right, which are making up the grana, and then you are seeing that the grana basically interconnected by each other through the lamella, right? So let's read the statements one by one. We know that it is not an animal cell at all. Why? Because we have already identified the chloroplast. It is a plant cell because it has a chloroplast. It is a plant cell because it has both chloroplast and nucleus. Nucleus is present in both the plant cell as well as the animal cell. So the correct answer and the most appropriate answer for question number 12 over here is option B, that it is a plant cell because it has a chloroplast, right? Let's move on to question number 13 over here. This question number 13 is from May June 2020 paper 1 question 5. The question says that the table shows a comparison between two features of a typical eukaryotic cell and a typical prokaryotic cell. Eukaryotic cell, you can say that animal cell, and the prokaryotic cell, you can say that they are talking about the bacterial cell, right? Now they are saying that which row shows the correct comparison between these cells. Presence of the centromeres. We know that the centromeres are basically present between the sister chromatids, right? They are basically joining the two sister chromatids together. So the chromosomes are basically present in the eukaryotes, right? So the centromeres are also present in the eukaryotes. In the prokaryotic cell has the linear DNA, right? And they do not condense to form the chromosomes. You can never find the chromosomes in a prokaryotic cell, right? So hence you cannot find the centromeres as well. Then if I talk about the telomeres are present at the end of the sister chromatids in the chromosomes, right? The telomeres are basically present at the poles of the sister chromatids of a chromosomes, and eukaryotic cells have the chromosomes, so yes, telomeres are present in the eukaryotic cell. However, if I talk about the prokaryotic cells, the bacterial cells, it does not have the chromosomes, hence it does not have the telomeres as well. So hence the correct answer for question number 13 over here is option B. Alright, so now let's move on to question number 14 over here. It is from May June 2020 paper 1-3 question 2. The question over here is saying that the electron micrograph shows part of a eukaryotic cell. Which cell structure is a site of protein synthesis, right? We know that the protein synthesis happens in the ribosomes, right? So we have to identify the structure of the ribosomes over here. So if I talk about C, so C is the centrioles, right? If I talk about B, so they are pointing to these dotted structures in the cytoplasm which are known as the ribosomes, right? And if I talk about these vesicles which are shown by the arrow in A, so they are basically the vesicles, or you can say that they are the lysosomes, right? Because lysosomes are also the vesicles containing the hydrolytic enzymes, right? So they can be lysosomes. So hence the answer to this MCQ would be option B, ribosomes. You just have to identify the ribosomes and mark it as your answer. Hence the answer is option B. Alright, so now let's move on to question number 15 over here. Now question number 15 is from May June 2020 paper 1-3 question 3. It says that some stains can be used to identify the cell structures in the living cells. A dilute solution of one stain causes the whole cells to appear blue. The blue color rapidly disappears in most cell structures. Those cell structures that release energy stay blue. Which type of cell structure is likely to stay blue? Alright, so they are basically saying that only those cell structures that are releasing the energy will stay blue, and they are saying that which is likely to stay blue. So we know that mitochondria is known as the powerhouse of the cell, right? It is known as the powerhouse of the cell. It has a role in the production of ATP. So hence the ATP is released from the mitochondria, right? We know that ATP is not released from the endoplasmic reticulum, it is not released from Golgi, it is not released from lysosomes, it is only released from mitochondria. So hence the correct answer for this MCQ is option D. Alright, so question number 16 is from May June 2020 paper 1-3 question 4. It says that a molecule of carbon dioxide is in the center of the mitochondrion. So let's draw a double membrane bound mitochondrion. We know that mitochondrion is double membrane bound. This is the molecule of carbon dioxide. This is the inner mitochondrial membrane. This is the outer mitochondrial cell membrane. Assuming that there are no other cell structures in this path, how many phospholipid layers will the carbon dioxide molecule have to pass in order to leave the cell? So basically they are saying that how many phospholipid layers basically the carbon dioxide molecules have to pass in order to leave the cell. For example, this is the cell, right? This is the cell membrane of the cell, right? So basically now there are three cell membranes. This is one cell membrane, the inner cell membrane of the mitochondria, the folded one. Then there is the outer cell membrane of the mitochondria, and this is the membrane of the cell, right? So we know that one cell membrane is known as the phospholipid bilayer, right? It is the phospholipid bilayer. So how many phospholipid layers will the carbon dioxide molecule cross, right? If this I am talking about just this cell membrane, so it consists of two phospholipid layers, right? Because cell membrane has a bilayer of phospholipids, right, which makes up the hydrophobic core. So how many phospholipid layers is carbon dioxide going to pass in order to move out of the cell? So it is going to pass six phospholipid layers. How six? Two of this membrane, the inner membrane, then two of the outer membrane, and two of the cell membrane of the cell, right? One, two are from one membrane, then two from other membrane, and two from other membrane. So hence a total of six phospholipid layers carbon dioxide must cross in order to leave the cell. Hence the correct answer is option C. So hence the chloroplast is going to have 70s ribosomes, the prokaryotic cells are going to have the 70s ribosomes, the mitochondria is going to have the 70s ribosomes, the cytoplasm of the eukaryotic cell is going to have the 80s ribosomes. Hence the correct answer for this MCQ is option B. Now in question number 18 over here, which is from March 2020 paper 1 to question 2, it says that which cell structures can form the vesicles. So yes, cell membrane can form the vesicle either by the exocytosis or by the and yes, so cell membrane can form the vesicle during the endocytosis, right, which is the process of ingesting the endocytosis, which is the process of ingesting the material inside the cell. For example, if this is a cell and this is a material which is coming, so if the cell membrane is going to engulf that material, right, so it is then going to convert it into the vesicle inside the cell, right? So cell membrane can form the vesicle. Endoplasmic reticulum, yes, obviously the endoplasmic reticulum, for example, if I draw a continuous rough endoplasmic reticulum over here, whatever the protein will be synthesized, it will be passed in a vesicle to the Golgi apparatus, right? So yes, endoplasmic reticulum can also form the vesicles. Golgi body, obviously Golgi body, the function of the Golgi body is packaging, modification, and we know that the lysosomes are also made in the Golgi body, which is the cell membrane surrounding the hydrolytic enzymes, right? It is a vesicle of the hydrolytic enzymes. So yes, Golgi body also can form the vesicles, as well as it can form the vesicles of the extracellular proteins which has to be transported out of the cell as well. So they all go in the vesicles to the cell membrane. So hence the vesicles are formed by all of the three: cell membrane, endoplasmic reticulum, and the Golgi body. Alright, so now let's move on to question 19, which is from March 2020 paper 1 to question 3. It says that the diagram shows three circles 1, 3 and 5. Circle 1, circle 3 and circle 5 representing chloroplast, mitochondria and the typical prokaryote. Now basically you have to identify that which circle is basically the mitochondria, what is the chloroplast, and what is the typical prokaryote, right? Which row correctly identifies the three structures and some structures that are shared between them, right? So basically over here you have to identify by the common structures, right? We know that the chloroplast and mitochondria has the circular DNA, correct, but however mitochondria and prokaryotes do not contain the 80s ribosomes, they contain the 70s ribosomes. So hence option A is incorrect. Option B is also incorrect, as they are saying that chloroplast and mitochondria has the 80s ribosomes, which is incorrect. They have the 70s ribosomes. Prokaryotes and mitochondria are having the circular DNA, which is correct, and mitochondria and chloroplast are also having the circular DNA, which is absolutely correct. Hence the correct answer to this MCQ is option C. Alright, so now let's move on to question 20. It is from March 2020, paper 1 to question 6. It says that which structures are found in a typical prokaryotic cell and also in a typical plant cell. So we can clearly see that cell wall is present over here, and cell wall is present in both the typical prokaryotic cell and the typical plant cell. In the typical prokaryotic cell, the cell wall is made up of the peptidoglycan, right? And in the typical plant cell, the cell wall is made up of the cellulose, right? So hence the correct answer over here would be option A. Alright, so now let's move on to question 21 over here. This question number 21 is from March 2020, paper 1 to question 5. It says that it is possible for a bacterium to synthesize a eukaryotic protein. This involves introducing a eukaryotic gene into the bacterial DNA. The eukaryotic gene is then translated by the bacterium. What explains why a bacterial cell can produce a eukaryotic protein but cannot produce a eukaryotic glycoprotein? We know that glycoprotein is made up of the glycogen plus protein, right? Glycogen plus protein. So it forms the glycoprotein, and this conjugation, or you can say this modification, is happening in the Golgi body. This modification is happening in the Golgi body, and Golgi body, we know that Golgi body is a membrane bound organelle, right? And prokaryotic cells, which are the bacterial cells, can do not have the membrane bound organelles. In Golgi body is a membrane bound organelle in which the glycoprotein is synthesized, or you can say that glycogen is combined with the protein, right? So the correct answer for MCQ 21 would be option D, that bacteria do not have the Golgi bodies, because these modifications happen in the Golgi body, right? Alright everyone, so that was it for today's class. In the next class, we will discuss the remaining MCQs of the cell structure. And if you guys enjoyed today's class and you like the past paper solution, do consider subscribing this channel, hitting the like button, and do share your comments that how do you find these lectures, are they useful to you, and which lectures or which topical past paper solutions do you want to see next. See you guys in the next topical MCQ video. Allah Hafiz everyone.